Not today. That's why I stand up in lectures. I don't want to fall asleep in the middle of a sentence. You sure can. All right, a couple of things. Today's Wednesday, so I think we all agreed that the exam will be next Wednesday. So I wonder if the United States will ever adopt what they do over in England, where you don't have exams until like your last year. I would love that, but as a student, I would hate that. I don't know. That's a lot of pressure for exams. But we do have to do assessment of some sort. So, should we do a review session again on Monday? All right. I will send out an announcement. I'll talk to you a little bit today about the exam content, but let me see how far I get today and that. So I'll do that toward the end of today because some of today's material will be on the exam. I did update just one slide in the plant-plant communication forested habitats. You'll see that I made a note that I updated it. I just wanted to show you what I forgot to be explicit on one thing that I wanted to be explicit on. So here we're looking, and this isn't the slide I edited, but I wanted to remind you that in this study that she was doing in the lab to look at the flow of carbon amongst plants that were in the pot, the donor and the receiver, we were talking about how some, the mesh in the pots that were surrounding the receiver were of two different sizes. One that would exclude roots and but allow mycorrhizal fungi to get through, and another that would supposedly not let mycorrhizal fungi, like the actual hyphae, to get through. And so that is here. So I put that in here already. We talked about this already. You know, hyphal connection was manipulated with the mesh bags. Seedlings were separated by a root blocking barrier that either allowed passage of the hyphae or restrict if it was the smaller mesh size. And then we were looking at some of the results, and I wanted to point out, so this was the one that I edited, slide 30, I think. So I added this second bullet. So mesh excluding hyphae still didn't inhibit transfer of carbon. And I don't know that I said that explicitly when we were looking at that. So I don't think they were quite expecting that, but basically what that's showing is even though the hyphae theoretically couldn't get through that, they were still exuding some of the carbon out into the soil, where then the hyphae on the other side of the mesh were picking it up. And so that's what they hypothesized, although they did talk in the paper that maybe the hyphae somehow like met up against the mesh and were able to more directly transfer if they were both pressing against the mesh on either side, or maybe the fungi degraded the mesh enough where they could get through. So they discussed several possibilities, but uh, their data was showing that even theoretically, though that mesh was blocking the movement of the hyphae, the carbon was still getting through. So either case, the mesh bags, whether they were bigger or smaller, you still saw transfer. Okay, so I just wanted to clarify that. And then I added this to overall, there's a diverse microbial community that was actively assimilating. And so that's a lot what this image is showing, that, the lion's share went into the fungi, but other microbes in the surrounding soil and on the rhizosphere were also getting that carbon, mainly because of leaky roots, leaky hyphae. And so they're allowed or able to capture some of that when it gets out. All right, so that's been updated. It's in the slide set that's been posted. Okay, then the other slide set I just posted a few minutes ago. Plant communication, kin, blah, 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 that one. So that one I updated considerably. Oops, okay. And wanted to get into in more detail today. Let me get this. Isn't that a beautiful picture? So that is fungi on the shoreline of a still lake, and you can see the reflection in the water. There's that lone little one off to the right. So they're not glowing themselves. That's the sunlight, the sunset, I think. The angle of the sunset was hitting them just right. But I have in the notes, this is called a saffron drop bonnet mushroom. That's the species. Saffron drop bonnet mushroom. What a lovely name. And they actually do glow very faintly. Not like that. It's more of a greenish glow. They bioluminesce. And so I took it from a little blurb about this discovery that they found. So, and I guess when they associate with trees and they're like the hyphae is growing into the roots or stem, they can show that the wood is actually bioluminescing too because of the fungal hyphae. So mushrooms are so cool. All right. So let's see. This idea now we're getting into, let's see, let's review a little bit. So we were thinking about plant communication. We've gone through this already. They started, looking at all kinds of ideas in terms of communication. Lots of arguments in the literature about what's a cue, what's a signal. We're not really going to get into that. They're still arguing about it, so there's not a whole lot to really say other than they're still debating, which is fine. And then, but you'll see words like this in the literature: exploitation, eavesdropping, manipulation, deceit. So they're really thinking in social contexts of what's happening when plants may be communicating with each other. And of course, they're looking at it from an evolutionary natural selection perspective. It makes sense that if you're going to send out some kind of cue, especially if it costs you something in resources, that you better be making an honest cue. And so this idea of looking at it from the framework of evolution and natural selection. So we went through this, looking at the idea that you should only be producing a cue when it gives you some chance of surviving and reproducing. And there's still debate on what is true communication. That is one idea of what I've seen, but again, there's debate. Last, we did talk about this slide too, this idea that, they're showing that there can be kin recognition. And so these started out as just some simple lab-based studies. Susan Dudley was really the one who spearheaded this research, looking at kin recognition. And so this idea that plants that were related, they didn't grow as extensive of a root system. So they were politely restraining themselves, is how they put it. And so when you think about a plant and they're in a pot with another plant, or just in a natural setting when there's lots of plants around each other, remember that plants are constantly assessing how much resource should I put into building my shoots and leaves? How much resource should I put in putting into root system development? And so they also, along with the shoots and leaves, is, how much should I put into flower production and all of that too, of course. And so we'll talk a little bit about this, a little bit more of, how a plant may be changing its body essentially in this idea of kin versus non-kin and how they're using that as evidence of kin versus non-kin. All right. We looked at how are they doing it. So we've already looked in the forest systems with Suzanne Samaran's work. She was looking at root exudates. But also VOCs when we looked at mountain pine beetle ideas and how you can have VOCs being spread from plant to plant. We'll look at red far red and blue light a little bit more, in not so much today, but later. And then we'll be looking a little bit more at ethylene and different hormones later. So it makes sense that you're going to have hormones involved in this whole cascade of recognition of kin versus non-kin, and then how to interpret that, and then how to respond appropriately. And so we'll get into that when it's necessary. Okay, so this is new information now. I wanted to talk a little bit about um, background. I've referred to this book before, The Secret Life of Plants. I put it up there really small because I really don't want you reading it. It's not really a good one to read. It's a lot of pseudoscience and false experimental designs, but the reason I'm talking about it is that it really had a major impact on botany research for decades. And it was published in the 70s, 1973. This is some of what I'm taking are quotes from an interview that Michael Pollan, if I don't know if you've heard that name, Michael Pollan. He's written a lot of good books about plants. And he was interviewing, and so these are some of his quotes that, it's a beguiling mashup of legitimate plant science, quack experiments, and mystical nature worship that captured the public imagination at a time when New Age thinking was seeping into the mainstream. So, in the 70s, this is well before most of you were born, all of you were born, I'm guessing. In the ensuing years, several legitimate plant scientists tried to reproduce the studies that were talked about in the book without success. Much of it has been discredited, but it's a little, the horse got out with the door open. What's that saying? The barn door. There's a saying, something about no sense closing the barn door when the horse is already out, or something like that. The book was already out, so it caught the attention of so many people. Lasting damage. So many plant scientists, I think anyone who knows about this book who's a plant scientist would say, this has caused so much problems, in the sense that it really dried up any funding. That was one of the biggest hits that plant scientists saw, is that the NSF and other granting agencies wouldn't take any grant proposals that had anything to do with plant behavior, plant communication. They're like, no, because of this book. And it really brought that field of research to a grinding halt. Well, Richard Carbon, which will be talking about his work with the sagebrush, he was being interviewed by Michael Pollan for this paper, or this article that Michael Pollan wrote. And he was saying how in the 80s, people working on the plant communication faced some of the same outrage that they're getting right now with plant intelligence. And so just simple, like, are plants communicating? That was a lot of censorship and a lot of harassment, essentially, in the science people's lives. He said it was enormously contentious. So Richard Carbon lived through this, and he was saying that, it's now generally accepted that, yeah, plants communicate. But back then it was very, very difficult. It took me years to get some of these papers published. People would literally be screaming at one another at scientific meetings. Plant scientists in general are incredibly conservative. We all think we want to hear novel ideas, but we don't. Not really. I think that's changing a little now. I see it more open-minded, but I still think, especially some of the older scientists, not me, but some of the older scientists that have really kind of held on to the idea, and they just can't quite open their mind to consider. And that's all it would take, is just consider it a little bit. And so this was also a very unfortunate situation. So David Rhodes was a scientist. He was a zoologist, actually, and a chemist that was employed at the University of Washington. And he started to look at a field study where he was noticing how these trees were getting attacked by tent caterpillars. And but over the years, they bunch of the trees didn't seem to get attacked anymore. And so that piqued his interest, and he's did a nice field study showing, and this is the abstract from the paper that was published in the 80s. And he basically was saying, yeah, it looks like, these plants are communicating with volatiles. The tent caterpillars don't seem to be able to be damaging them anymore, even though, they weren't initially chewed on. So something, a tree that's so distant that roots couldn't have been interacting with that, the root systems. Nope, it couldn't have been that. So it's probably volatiles. And his data was very well done. His experimental design, his data collection, and all of that. So he was super excited about this, and he went to conferences and again, screaming at scientific conferences ensued. His peers said, no, this isn't correct. This can't be happening. And this went on for years. Then, when he tried to reproduce, so of course, that's one of the big things, right? If someone publishes something, if you really want to verify it, some other group, or they should redo it to, see if it really is legit or if it was just a one-off. Well, he had problems with future research on that. He couldn't always reproduce his initial findings. Well, that was like really bad because now he's being discredited. He basically discredited himself by saying, hey, I can't replicate. That was really unfortunate. What he didn't know, and they later found out, is that he was doing his reproductive, when he was trying to reproduce that experiment, he was doing it in a totally different season. His initial study was in the spring. When he was trying to reproduce, I think he was doing it in the fall. Yeah, in the fall. And plants behave very differently under those seasonal shifts. And so that's why he couldn't reproduce it. Now that's like common knowledge among plant scientists, but back then they didn't know that. So very unfortunate. He just had a lot of bad luck. No one would, the granting agencies wouldn't touch him. If he submitted grants, they wouldn't give him any money. So that put a halt on his research. So then he turned to mentoring students and young professors because he said they were more open-minded to some of the ideas that he had. And one of the persons that he interacted with was Richard Carbon, who was younger and had just got his PhD in entomology. So he mainly studying bugs. And when Carbon was teaching a graduate class, he invited Rhodes to give a talk. And then they met and then they started to collaborate and talk about their research. But by then, um, Rhodes, he stopped all of his research. He went to a community college and taught. And then he ended up buying a motel and running that. And then diagnosed with cancer and died in 2002, well before some of this research that we're talking about came to fruition. And so he really had a series of really bad luck and a lot of community shunning almost in terms of his ideas were just not being considered. And he was getting yelled at a lot in conferences. Well, then two other scientists who I'll mention at least one of their papers, Ian Baldwin and Jack Schultz, they were at Dartmouth College. Right or soon after he died, they published similar findings to what he had found. And that timing, the fact that it was, after the 1980s, and the fact that it was a lab study where Rhodes did a field study, which is a lot messier, but they did a very carefully controlled lab study. And initially they did get pushback from the scientific community, but it was short-lived. And so now they're both very active scientists. They're very successful scientists working in this field of plant behavior. And when they did publish, they did cite David Rhodes as like the first person, the pioneer scientist who really broke open this field of research. So that's the actual publication. So it's kind of a sad story from David Rhodes' perspective, and it just goes to show that timing can be everything. And that ridiculous book that got published in the 70s, that was perfect timing for what the general population was doing with culture. And so bad timing for a lot of reasons, led to that all of this research being quashed until relatively recently. Well, now Richard Carbon comes, he's been building on his research, and so many other plant scientists refer to him as, very rigorous. He's very well respected in his field. And he has been doing long-term ecological research in a pretty decent field of sagebrush near Mammoth Lakes, California. And he has had this site now. I think he's been working on this site for over 30 years, studying it. So they know a lot about it, collected an amazing amount of data. And what's really interesting is by the time he got into university research and teaching, he was able to talk about plant behavior openly and have it be accepted. Plus, he had a reputation for being very rigorous, so that helped. And he actually started turning to people who study animal behavior. And it's kind of similar to what Monica Gagliano has been doing with what can we learn from animal behavior research design? Can we take that experimental design and apply it to plants in some way to ask if they have certain behaviors? So he's been doing this for many, many years. And one of his earlier research, when you start thinking about kin recognition, well, in order to recognize kin, you have to kind of self-recognize yourself. You have to have self-recognition. So that was one of his earliest papers with a colleague from Japan. And so he said, we really do need to explore this idea as we think about kin recognition. And so he, again, he's pointing right to the animal behavior world and thinking about that. And then he starts looking at, it could self-recognition be going on in plants? And like I said, his plant of choice is sagebrush. And they already knew that they are producing volatiles. And those volatiles can be, used as signals to neighboring plants around. And he says, here we show that plants that received volatile cues from genetically identical cuttings. Okay, genetically identical. So he was propagating them. He was taking cuttings from like the mother plant and using those cuttings as the genetic identical cutting. They accumulated less damage than plants that received cues from non-self cuttings. So the nice thing about plants is you can propagate them clonally and keep them alive. So this was a great start to the research. And he is saying, they were able to tell this non-self discrimination didn't need physical contact. It was through volatiles. So this is the experiment that he did. So he has this parent X that he took cuttings. So cloned means he propagated through cuttings. Okay, so he has those in separate containers. And then this is what the second line down there is the assay. So treatment one was no clipping. Okay, so they didn't expect any kind of defense response for that treatment. Treatment two was they cut. That's a lot of what they do. They physically take scissors and make cuts. And they snip off a portion of the branch. And then they assay. So the assay just means then they take a sample after they've clipped. And because it's on the same plant, they should see a nice induction of defense response. Then they said, let's take the parent X and the cloned cutting, so they're identical, and do the same thing. Let's make a cut and then assay the genetically identical plant next to it. More induction than non-self was the hypothesis. And then here in this last treatment 4, they had two they weren't related. They weren't genetically identical. And they clipped and then assayed the neighboring plant. So nice design. Here's some of the data. So here you see the just follow down all the way down to the x-axis for both of these so you can see what's what. So these are different years. 2007 is on up top and 2008 on the bottom. So your control, you can see, has the most damage. So this is the left, both in 2007 and 2008, you have lots of damage. Those are the ones that weren't clipped. All right, that's just the controls. And then you can see here, treatment 2, you see less damage when the plant was clipped. And then 3 and 4 is really what we're zooming in on here. So comparing these two, you can see that if it was genetically identical, you had significantly less damage compared to if they were not genetically identical. So there's a difference in response depending on if they're genetically identical or not. And that is the data that they were pointing to in order to say plants are able to recognize self versus non-self. And they said, if you're going to be looking at this idea of kin versus non-kin, before you can even do that, you have to ask, can they recognize self from non-self? That's the first step. So that's what the second bullet point there is getting at. So this study was very important. It kind of set the groundwork for doing work on kin selection now. I did not post this paper, but please be familiar with the experimental design idea, like what were they doing here? How did they test it? So please make sure that you're studying through this. The reference to the paper is in one of the notes section if you really want to look at the paper, but I'm not assigning it for mandatory reading. Okay, so that self versus non-self publication was a good first step. Then kin selection theory has been around for a long time. It's not like it's something new. In the 1960s, Hamilton came up with this idea as a way to explain altruistic behavior. And so this is kind of so we're looking at animals and we're thinking about this idea that, they're going to cooperate with each other. It's more likely they'll cooperate if they share genes or the same alleles, if they're really closely related, especially when times get tough. You'd think, yep, they're going to cooperate with each other if they're closely related compared to if they're not closely related. And then getting into this idea that, natural selection should favor traits that, increase the reproductive success of relatives. And so you will favor your kin because then at least some of your genes get into the next generation because you're closely related to your kin. So you're going to favor your kin, even if it might make you take a hit in some way. All right. Now, this is especially kind of easy to think about if you think about conditions where resources are limited, scarcity of food, scarcity of resources of some sort. It was really Susan Dudley's group that published that paper on the sea rocket plants that had some nice data to show that there seems to be kin recognition going on with the sea rocket plants. And that was the one in the video that I asked you to watch for the first exam, and what we were just talking about a few minutes ago, this idea that they would politely restrain their root growth if they were growing in the same pot as their kin. And so they weren't overly competitive with each other for the mineral and water resources in the soil. That publication is often pointed to as the start of showing, yeah, there could be something here. This is taken from a review paper that talks about the different definitions. Please be familiar with these definitions. I think most of these you've probably encountered in some of your science classes if you've taken any kind of ecology classes. Not sure if facilitation has been something that's been talked about with you yet, but all those definitions are nice, nicely put. Inclusive fitness. Yeah. So get comfortable with these ideas. We'll be talking using some of these terms as we work through what's going on here. So this is the paper, the Dudley paper, that was getting at the kin recognition in the sea rocket plant. And so this is the kind of the summary, the abstract of that. So this last sentence, because greater root allocation is argued to increase below ground competitive ability, the results of the restrained root growth are consistent with kin selection. Plant scientists have been thinking about this quite a bit. Like what could be going on? What could we study to get at kin recognition in plants? This figure is trying to get across some of these points. So if you kind of look over the where it says signals or pathways of kin recognition, so all of those underneath that. So there could be volatile signals. We've definitely talked about that. Light profiles, we will talk about that. That's the red far red ratio or the phytochrome pigments. Cryptochrome. We'll talk more about that. Root exudates, we've certainly mentioned that, and then the common mycorrhizal network. So those are all ways that signals could be sent that at least these authors were thinking about. We now know there could be sound added into the mix, as well as some other ideas. And then on the left here, they're saying, okay, what could be the effects on the plant if they can tell the difference between kin or not? And they mention a few here. What they've drawn, so these two, these are the kin, and then they have a non-kin next to it, okay? So what they're doing is trying to draw out like what could be some benefits if you can recognize your kin. So more attractive to pollinators. If you have kin nearby, you kind of have, you're very similar. You have similar flowers, you have similar pollinators and pollinating mechanisms. So you can imagine that this could be good for getting this phase of your lifestyle to completion where life cycle where you need to get pollinated. So yeah, you can imagine a cluster of kin hanging out together. The pollinators might hone in on that better. That reduced competitive intensity, again, restraining root growth. They're trying to show that there is a difference compared to over with the non-kin. And then more investment in the common mycorrhizal network compared to if you're non-kin. We'll talk a little bit more about that common. You see they have a typo there. Should be CMN. The other side, okay? That says CW then, doesn't it? Okay, I just noticed that. Huh. Oops, typo. All right, common mycorrhizal network. All right, so we'll come back to this. Some of these ideas, I mean. All right, so here's another paper with Carbon and his collaborators. And this is one of the earlier studies. This is 2013. Kin recognition affects plant communication and defense. So they were choosing to look at how do plants communicate if they're getting attacked or if they're injured, which in their, if you cut them with scissors to the plant, it's like, I'm getting attacked. So Carbon went back to his sagebrush fields. So Carbon does pretty much all field-based work, which is messy, a lot of variables. I give him a ton of credit, but he's got a fantastic field plot of all these sagebrush that he studied so intensively. So he's got a really good system. So here they have this idea where they take a plant and depending on if they're close relatives or distant relatives, how are those plants responding when they clip the plant? Excuse me. And so the data is being showed below there. Damage on the y-axis. So when they clip the plant, cues from the distant relative, you can see they got a lot of damage compared to if they got a cue from a close relative. So very obvious difference. So basically what they do is they take scissors and make a clipping and then they put a bag over the clipped branch and let it sit there for a while and accumulate. And then they have these huge, I like to see a 1 liter syringe. That's big. So they take a 1 liter syringe and suck out the gas and the volatiles in the bag, and then they transfer it over to whatever plant in the close relative or the distant relative. And so this idea of um, looking at the damage. So what they came up with is that there's variability in terms of what are the volatiles. But they did definitely see a positive correlation between genetic relatedness and the similarity of the volatiles between those really closely related. And so this is kind of getting at the chemotype idea that we mentioned last time, that if you're genetically more closely related, you tend to have the same chemotypes. In this case, it's what type of volatile compounds, what secondary metabolite profiles do they have as a volatile. So because they're closely related, it kind of makes sense that the kin can pick up on that because they produce similar compounds. They're like, oh yeah, I understand that, and I'll respond to it appropriately. And they also pointed out that if you do have that variability in volatiles, it kind of keeps conversations private because you can have a very specific chemical that you can send to your kin and only the kin can understand it. And then other plants will have a different chemotype or a different type of volatile compounds. And so there's like these private conversations, so to speak, that could be happening. So that was one of the ideas that they were talking about. Okay. So that study was very good at looking at the idea of kin versus non-kin. And that drove them to do some more research on the chemotypes, looking at what's really going on in a more fine scale here. And so they said, yep, volatiles are important for sending out cues to, ramp up your defenses. You're next, you're going to get attacked. And so then they looked at volatiles from different sagebrush plants and used GCMS to analyze those. And they were finding, yeah, those profiles really are highly variable. And so they started talking about different chemotypes based on their chemical composition. And that it's, related to the genetic mechanisms. And again, so this is really reinforcing that previous study. They just were going a little bit deeper into studying the chemicals, the chemical nature of the volatiles. Okay, so that was backing up their research from the previous paper. Well, then part of that field plot has a lot of wild tobacco. And of course, that's not sagebrush. So they were looking at, is wild tobacco doing anything out there or not? And so they were looking at how tobacco might be responding to any of these VOCs that are getting emitted. And so this was published in 2001. So this is earlier research. And they are finding, so it says right up there that the sagebrush, when they clipped the sagebrush, the wild tobacco did become more resistant to herbivores. 15 centimeters, so it's kind of close. Yet they had to be fairly close to get it. Another thing I want you to pick up on here, let's see. Associated with lower levels of herbivory were increased activities of polyphenol oxidase in tobacco. So they can track polyphenol oxidase being produced by tobacco and use that as a proxy to, response to herbivory. Okay. And they did see that they had increased PPO, so polyphenol oxidase, they abbreviate down to PPO. So saying, yep, the tobacco were increasing PPO when they're near the clipped sagebrush. So now we're talking about interspecies, species between two different species of plants, and that there was communication going on in this respect. 2001, they said we got a lot to learn here. But a later study, or no, is this? No, never mind. Okay. So then they went deeper into it. Here we report the first rigorous, he even uses that word, rigorous experimental evidence demonstrating that undamaged plants respond to cues released by neighbors to induce higher levels of resistance against herbivores in nature. Sagebrush that was clipped released a pulse of methyl jasmonate. So remember, jasmonic acid or jasmonate's a plant hormone, but when they methylate it, it becomes volatile. So they started to hone in on that. They're like, huh, I wonder if methyl jasmonate is the signal. And they were measuring the polyphenol oxidase in the tobacco. And they were actually tracking grasshopper and cutworm damage after they cut the sagebrush. Then they started to look at how many grasshoppers and cutworms visit those tobacco plants that are near the clipped sagebrush, and what are we seeing in terms of damage. They even looked at this idea of, is it VOCs only or is it root contact as well, or mycorrhizal contact? And so they did a treatment where they looked at that too. And they said blocking the soil contact between the sagebrush and the tobacco didn't have any effect. So basically, they were coming down to the idea that it was the volatiles that the tobacco was responding to. So PPO activity, that's the tobacco plant. Okay, so they're tracking, that's its defense mechanism, one of its defense mechanisms. And so when it was near a clipped sagebrush, you could see its PPO activity went way up. So it was starting to defend itself because of the sagebrush getting damaged. They followed it out for 3 years. Really pretty data. I mean, look at how consistent that is. Unclipped versus clipped, proportion of damaged leaves. That is really good data to see that kind of consistency. And then the damage. C is maximum number of leaves. by cutworms. So these were the cutworms in figure C. I think figure B was the grasshopper. Yeah, grasshopper. So really consistent among two different predators. So, I mean, really pretty data. This is amazing. So this idea of this is where they blocked the air contact on the top. So they said, what happens if we block tobacco's ability to get any access to the volatiles? And you can see pretty similar bar graph results in terms of the damage done. And so that's basically saying, yeah, if they can't get access to the VOCs, they experience roughly the same damage across the board. But if you block the soil, you still see a pretty good difference. So that's evidence that it's VOCs and not soil-based information going from the sagebrush to the tobacco. They mentioned methyl jasmonate in their paper, but they didn't want to commit. So they're like, we think methyl jasmonate's playing a role in it. And now we know that methyl jasmonate is released by plants and other plants can respond to it. We do know that now, but this was an earlier study where not enough research had been done. So they were tentatively suggesting methyl jasmonate. Okay, but really nice data, especially when you consider this is a field study. To get that clean of data is pretty amazing. Any questions on some of the carbon research? Carbon? So he's the sagebrush dude, and he's still at it, apparently. He's still doing that work. I did put one of his papers into EDUCAT, but he's published like 200. So I just put one. Okay, why don't we take a break because the next topic is kind of switching gears a little bit. So let's take a break right now and we'll come back at right around 3. All right, part 2. So now we're getting into what about mutual benefit hypothesis? How does that compare to kin selection? The data in that's coming out with all the research with Richard Carbon's work and others are showing that kin selection can happen amongst plants. It's, a little messy. Sometimes we see um, behaviors that are a little different than what they expected. So it's not 100% clear cut, super, like Richard Carbon's work is very nicely laid out, but some other studies has shown weaker connections to this idea of kin or have even shown the opposite. And so it's not 100%, but the data is piling up that there's many, many plants that have been tested. I think, well, one report, the lead author said over 50 plants are known to do this kin selection idea. And then in another paper, I saw 30 plants. So it depends what year it was published, but you can see that it's not just sagebrush where this is happening. And so with the kin selection hypothesis, we've already covered that idea that if you're closely related, that you will probably use some kind of private communication to your kin. And that's going to benefit, you overall, your species overall, this inclusive fitness idea. With the sagebrush, he was showing that you can have chemotype dependent VOCs being released that kin to kin can understand each other, but kin to non-kin, not so much. So this was a good demonstration of it, kin selection theory or hypothesis. But there's this other idea, the mutual benefit hypothesis, where information is exchanged more freely between individuals and they don't have to be closely related. So kind of like that sagebrush and the tobacco idea. So basically, if you do, think about real world where plants are living together in a patch of field or forest, that pretty much all of those plants are getting the signal, and even though they're not related, they're responding to that signal appropriately. Okay, so these are kind of like very different ideas, and they've been debated about in the literature, like, what could go on here. One of the nice studies that was done was actually done by a Finnish researcher and his collaborators. This was done in 2019. And the way they phrase things, a little, I don't know, to my brain, it's like a little complicated, but basically, the paper is saying, yep, VOCs are being used as a way to transfer information. And now they're saying herbivory, so the herbivores are actually a selective pressure on the plants that is selecting for the plant's ability to share information with neighbors. Convergence on common volatile signals. Volatile signal characterizes information sharing phenotypes. So basically what they're talking about here is, yep, they're using VOCs. It can be a kin to kin, but this convergence idea is saying, now we're talking mutual benefit idea, all right? And then the little brief summary statement there. Chemical information transfer between tall goldenrod plants evolves in response to natural selection by herbivores. Herbivory selects from more open communication channels. So that's talking about the mutual benefit idea. They just don't come out and say it in this. And so sometimes scientists do that. They kind of dance around it and won't actually get into it too much. So these are goldenrod plants. So Richard Carbon said, okay, he must have either talked to these scientists or read their paper. This is some of I didn't post the paper, but I do want you to read through what I've posted here on the summary and the results and discussion. And so they are getting into, why they were studying the VOCs, because they are used for signaling. And talking about neighboring plants could also respond to those cues and then ramp up their defense against, oncoming herbivory. Here we test the prediction of two alternative hypotheses, the kin selection and mutual benefit. So they say it in their abstract. To uncover the selective environment that may favor information sharing. So they were interested in, is there a way to kind of create a situation that we can demonstrate different kinds of sharing of information in the plants, in the field study? So they did goldenrod. Basically, what they talk about down here now is the idea that if you have herbivory going on, you get more of the mutual benefit hypothesis, where the signal is getting out in a more common signal, where more goldenrod plants can understand it, so to speak. Okay. This is some of the results and discussion. I just clipped part of it, not all of it, but where they are looking at their data, talking about, this idea that neighboring plants tune in on the cues for, kin recognition, eavesdropping. It'd be kind of good if you could eavesdrop, because then you could gear up your defense ahead of time and experience less damage. And then they get into the mutual benefit idea that if you have a lot of herbivory coming at you, that then the mutual benefit idea might be coming into play. You can imagine that if there's a lot of pressure lots of grasshoppers or whatever coming at you, that maybe you want to warn everybody, not just your kin. And so these are some of the research ideas that they're trying to get at by setting up these experiments and looking at is, when does kin selection seem to be playing a role? When is mutual benefit playing a role? Are they mutually exclusive? Can they be both happening at the same time? It's a lot to try to tease apart. But this was a really good stab at it. This was like one of the first ones looking at this more carefully in a designed experiment. And then Carban came along and said, let's look at it. Let's play around with the sagebrush field studies that he has access to and has been studying. And so this is what, kind of what they were testing, looking at what happens if you give them low herbivory pressure where there's not that much going on. There's some herbivory, but not much. And then what happens if you give them a situation where there's really high predation? Is that going to influence whether they turn on this kin talk where they only talk to their kin, or where they broadcast more openly to everyone in the area? This is his abstract to the paper. What selective mechanisms favor plant communication and what conditions allow individuals to benefit by both emitting and responding to those. And so the kin selection and mutual benefit. How can we tease those apart? As predicted by the kin selection, we found several uncommon chemotypes, with some chemotypes occurring only within a single population. So it's like they have their own private language. But consistent with the mutual benefit, phenotypic diversity was negatively correlated with herbivore pressure. Negatively correlated. So that means that there was low chemotype diversity in those cases. Sites with higher levels of herbivory were associated with a few common cues, broadly recognized by most individuals. So, whoops, that I was underlining, not trying to cross off. So he's getting at there's both going on. It depends on the selection pressure. Do you have low herbivory or high herbivory? And that seems to play a role in whether or not they're going to just do kin selection and favor their kin and send a very specific chemotype to their kin that only the kin can figure out and ramp up their defenses, or with really high herbivory, go ahead and just, hey, we're all under attack. Let's ramp up. And this is another statement. The privacy of alarm signals is dependent on the risk level and relationship. Warning signals may be as subtle as gestures between kin, or as public as loud vocalizations that are perceived by conspecifics and heterospecifics. Do you know what con and heterospecific means? Conspecific means they're related, same species. Heterospecific is different. They have to be so fancy with their words. So I like the analogy in this later case where you shout it out to everybody. Alarm signals may converge as seen in mixed flocks of birds. So when you have mixed species of birds in a flock, if there's a lot of danger, they'll send out an alarm that everyone, all the birds, can understand and respond to. So if you can use a volatile that is kind of universal, a universal language, so to speak, when there's lots of herbivory. But when you want to just talk to your kin, then you use a very specific language, not a universal language. I often think when I'm reading about this kind of stuff, my dad was 100% Finnish. He grew up in a household where his parents only spoke Finnish. And so this idea of you speak Finnish, close kin, but when you go out of the home, they were forced to speak English because they lived in America here, and you actually got hit if you spoke Finnish in school. So switching to the universal language of English and where everyone can understand. That's kind of what I think about to make sense of what they're getting at here. So basically, this idea that different languages are being used, different VOCs are being used. Alarm calls of sagebrush converge when herbivory is high. Again, that's just saying you use a common language, a universal language when you have really high predation. And so this is another paper that was published just recently. Um, whether or not to listen in on some of these cues. Where is it? Previously, we observed that sagebrush populations that were in locations with high herbivory exhibited little diversity of their volatiles. So that's that universal language. High risk of herbivory could have selected for individuals that converged on a common alarm cue that all individuals would respond to. Alternatively, low chemotype diversity would allow higher levels of damage to plants. Let's see, rare. In this case, oh, here it is. In this case, rare chemotypes would experience less damage than common chemotypes. So they're going through again, is it kin? Is it the mutual benefit? And so in their paper, they said, we are seeing chemotypes that are very rare when you have this kin selection idea but when the herbivory goes to really high risk, high predation, then they switch to a very kind of common VOC that everyone could hear and understand. They mentioned the goldenrod study that was published before them that we just looked at. And they're saying our study suggests similar processes at the landscape scale and that herbivory may contribute to shape plant communication systems. So, basically they're saying it's very context dependent on whether or not you're going to get this association with kin and only kin are talking to each other versus, a mutual benefit. Let's shout it out to the world. So instead of the so-called coded whispers that they might use among just their kin, like just speaking Finnish in the house, broadcast the threat over the loudspeaker. So if, my dad, if that neighborhood was on fire, they probably wouldn't come running out of the house saying fire in Finnish. They'd yell fire in English to get everybody out of the neighborhood and get going on it. So that kind of idea, broadcast the threat. Use chemical phrases easily understood by all. Because really, I mean, you don't want to be left alone in a field. If you're a plant, you don't want to be the only plant left standing, So if you can defend yourself, it's coming and you can defend yourself ahead of time, but it doesn't really do you any good if you don't warn everybody when the predation's really high. If you don't warn everybody, then you're alone and you have no one to mate with. Pollinators are going to come looking for you, very, very rare. And when you think about it, there was one scientist was being interviewed and saying, this really is kind of the hypothesis to demonstrate intentionality, that they can put out these signals that are meant to be heard either just with their kin or to everybody. And so it really does get at this idea of intentionality, which then some scientists would say that's an indicator of intelligent behavior, which is a whole nother discussion that we won't get into right now. So what they were discovering in the plant world, they actually have seen already in animal behavior, that songbirds that we were talking about, where, flocks of birds, if there's low predation risk, they use a certain song that's only to their kin can understand. And then they've shown through careful studies that when the predation risk is really severe, then they make an alarm sound that everyone can understand, even different birds in that flock. So that's been documented. And now when you look back at what's going on in the plant research on this idea, basically it's like similar to the idea of dialects. You know, the birds have their songs, the plants have their dialects of, in this case, the VOCs. And it's also showing that communication is very complex and layered. Sometimes it's for kin, sometimes not. A lot of things to think about in the environment, what could be influencing how they're going to communicate. You know, we've been focusing on damage, herbivory damage, whether it's low or high, but there could be other things going on too that would influence how these plants are communicating with each other. So it's really kind of opened up this whole idea of plant communication is way more complex than we thought, and it's layered, and it is, you know, definitely a situation where they can recognize their themselves versus others, kin from non-kin, and they can adjust their communication appropriately. So this has gotten into um, some really interesting ideas about plant personalities, which sounds weird, right? Like what? Plants have personalities? Just kind of a easy way to talk about it. But Richard Carbon, with all of his work that he's been doing with those sagebrush fields that he's been doing for over 30 years, he knows every single plant. He has them all genetically sequenced. He knows so much about them. And so he started thinking about this idea with this idea of kin selection, mutual benefit. Sometimes, like I said, the research is a little messy. It's not like exactly consistent. And so basically, when he has been studying over these last couple of decades, you know, he has seen variation among the individual sagebrush plants and how they respond to whatever experiment they're doing. And if you think about animal research, animal behavior research, the plant personality idea, you know, this idea of how brave are you? How fearful are you? Are you a risk taker or are you generally risk averse? And so we all, you know, it's pretty easy to think about that with human behavior, that we do have that type of personality. You know, we have personalities that makes sense. Well, then they said, what about other animals? And really, people didn't start really thinking about that until just about 20 years ago, which seems a little late to the game, but you know, we are very human centric. And so we like to think that we're above everything. And but then when they finally said, hey, I wonder if other animals might have personalities. And so there's a really, whoops, 2017, not 2107. She didn't time travel. So in 2017, Charlene, I don't know how to pronounce that name. Anyone know how to pronounce her last name? Couchot? I don't know. We'll go with that. Couchot. She contacted Richard Carbon and said, hey, I have to do a special project for my PhD where I work outside of animals, but try to do something with behavior. And he said, great. And her study was on chipmunks. That's what she studied at the Vermont-Quebec border. And she noticed that chipmunks definitely have distinct distress calls depending on if they're getting scared by something that's flying over them or if it's something chasing after them. And she, it was quoted in a interview. She said, yeah, some chipmunks would be eating seeds and a leaf falls on the ground and they panic and make a call, screaming their little heads off, thinking it was a bird of prey. And so she labeled them as shy chippies. And then the other ones, and then other ones nearby would just go, you know, like, yeah, whatever, and they'd keep foraging. And so she called those the risk takers. And then she noted, you know, it seems like, you know, if those shy chippies are crying wolf all the time, ee, we're getting attacked, we're getting attacked, maybe you shouldn't trust them. It's kind of like the boy who cried wolf, right? And so then she actually developed a continuum of shyness to boldness with all these chippies she studied. She got so good she used earrings, different colored like earrings to label them or not label them, tag them. And after a while, she didn't even need that. She just knew them so well just by studying them for so long. So she really got into it. And one of the interesting pieces of her research was showing that chipmunks paid more attention to the distress calls from a really bold chippy than a shy one. They were kind of dismissive toward the shy chippies, like, yeah, you say that all the time, and you're always just scared, and I'm not going to really stress myself over that. So anyway, they started communicating, Charlene and Carban started communicating on what could we do as a joint study. And so they started looking at this idea in the sagebrush. And Richard Carban says, hey, maybe there's something analogous we could play with here. And he knows those plants so well, kind of like how she knew those chippies so well. He knows his plants so well that they started working. And he did notice that, when some plants signal a distress, VOC, that sometimes the kin neighbors, they wouldn't necessarily ramp up their defense or they wouldn't produce very much in certain individuals. Okay, so there's some messiness to the data, like in any study, especially a field study. And so he was questioning, could this be because these plants have different tolerances to risk. And so that's why they showed a little bit different response. And or maybe it's the emitter was a scaredy cat, like in the Chippy study, and the kin are like, yeah, whatever. I'm not really gonna put resources into making these defense compounds if I really don't need to. And you might just be a false alarm. He also noticed on all of his studies that, they prefer to get intel from family members and they don't trust, so to speak, the genetic strangers as much. I mean, that's kind of common to human behavior too, as well. So, but when you look at how research is done in, especially, botany, you take the average of all of your samples. So if you have 60 sagebrush plants and you're looking at how does this treatment affect root growth, you pool all that data, take an average. And that's legit. I'm not saying there's anything wrong with that. It's just that if there are any outliers, it's something that's off of, well off the data point, you see it's way over here when everyone else is over here, and you're like, oh, that's an outlier. And those make us uncomfortable when you're analyzing the data, and often it gets discarded. Like, oh, that's just too weird. We're just going to forget about it. And so now he's saying, hey, maybe that's because that outlier has a different personality, so to speak. And we should really be looking at individual differences and not lumping and taking an average. So this is definitely more challenging research. But like I said, his sagebrush experiment was pretty, or his field plot, he has studied that for so long that it wasn't that hard for him to get into this angle of let's study the plant personality idea, especially with the work that with Charlene's work and Chippy work. And so this is the paper he just recently published a couple years ago. And it's actually amazing that this didn't get totally discredited. I mean, look at that. Do plants have personalities? That's a pretty bold statement. I'm I'm going to do one more search to see if that kicked up some dust because I would think a lot of scientists might go, what in the world are you doing? But on the other hand, Carban is really well respected. And he had a lot of luck early on in his research program. So a little different than Rhodes, unfortunately. All right. So he's saying he always points, to animal behavior, saying, hey, we got legit research going on with animal behavior, animal personalities. So we use the term personality to refer to intraspecific expression of behavior that are stable over time and consistent across different situations. Animal personalities have been found, and it could be meaningful. So he said, okay, let's take a peek at what sagebrush are doing. So they were looking at doing individual plants within a population vary consistently in their responses to cues. Do the individual plants consistently vary to the different receivers? Are these stable over time? And are they effective emitters? Are they also effective receivers? And so these ideas that they are testing, he's saying if they meet this criteria, then we can say that they have their own version of a personality. And so I'm not going to get into the research. It's pretty gnarly and we don't have time for that. But um, this is part of the discussion. And levels of damage from the herbivores. So it's the same idea of clipping and then looking at response to that. Levels of damage from herbivores differed among individuals in the population. The various branches on individual receivers exhibited characteristic levels of damage that were consistent after receiving volatile cues from different clipped emitters. Indeed, the identity of the receiver explained a majority, 64%, that's huge, of the variance. So basically, that's really good data saying that the experiment, so basically, the experiment did back up all four parts that they were studying. The data did show that these individuals were behaving consistently over time, and we're displaying what animal behaviorists call how they define personality in animals. So now he's saying we can now think about this in the plant world. So this was just published a few years ago. And like I said, I'm shocked that this hasn't met with a lot of screaming at conferences. So I'm going to take a peek and see if there's any naysayers, but I couldn't find anything right before class. So this is really kind of fun to think about. So he's saying these outliers, they may have their own little personality. And then this is in their conclusion section, why does it matter? And just like in animal research, the fact that animals will behave a little differently with respect to other members of their species, it's important. It affects the evolutionary trajectory. It affects so many of the ideas of their successfulness at getting resources, surviving to adulthood in order to mate and get your genes into the next generation. So they're saying, yeah, this does matter. We should be looking at it. It's something that is important to think about the evolutionary process of these different species, including plants. The idea that individual plants may show consistent tendencies that can be quantified leads to a different research focus. So he's saying this opens up a new field of study. In the Bayesian jargon, the existence of personalities means that informed priors can be used to improve predictive power. So basically, past behavior often predicts future behavior, is what they're getting at there. Okay, so really interesting ideas being put out there by some different scientists, and the overlap with animal behavior versus plant behavior is getting tighter and tighter. What do you think? Do plants have personalities? How many people have worked with data where there's outliers? Yeah, those pesky outlier pieces of data. And it's like, what do we do with this? And he's saying, don't ignore it. Could mean something. So, okay. All right. So give that some thought. Read through those sections that I've took screenshots of, and I didn't post these papers because they're quite involved. I just took the take-home messages essentially that you need to know for these concepts. Where is the other thing? Okay. I think I'm going to stop there with, I have some other stuff to talk about, but I think I'm going to stop there because I wanted to talk to you a little bit about the exam for next week. So we're kind of squared away on what we got to be focusing on. let me. Okay, I'm going to get into EDUCAT so I can show you. Okay. All right. So this is the PowerPoint section of EDUCAT. So we finished off with epigenetics on the first exam. All right. So essentially, we're going to start with the bioacoustics slide set, and then the plant mimicry and vision. I'm not going to ask you about plant versus octopus. Just interesting to think about, but you don't need to study that. However, signals from sunlight and plant-plant communication with forest habitats, and then what we covered today in the kin, non-kin, all of that. So what we covered today goes through slide 47 on that slide set. There's a handful of slides beyond that, but we'll save that for next time. So it's essentially 5 PowerPoint slide sets, I believe. Yes, 5 slide sets. Then readings and videos. Start with the journey of sound to the brain. And then there are a couple of papers with bioacoustics. This video on the vibrating bumblebee is something that you might use as a piece of evidence to support whatever you're trying to tell me. So it's not like you have to study it, but maybe using it as an example to shore up what you're talking about would be appropriate. This paper I didn't talk about, but I do want you to read it and understand it and know the basic idea of what's going on here. Some of the concepts in this video by Stefano Mancuso are relevant to what we've been learning, so they can back up. You can use it as pieces of evidence to some of your answers you might be providing. You don't need to study this TED Talk with Dr. Gagliano. I don't know if any of you have had a chance to look at it, but it's kind of going along the idea of what we talked about earlier with David Rhodes, just being persecuted for, him saying that plants can communicate. And so she talks a little bit about how she's been persecuted in the recent history here, but you don't need that for the exam. Then there's these readings, and they're short for the most part, and we talked quite a bit about them. So a lot of the slide set is summarizing this work. So it's not as big of a deal as you think, but you do need to be familiar with these concepts and use these readings as backup to what you're learning through the lecture slides that we've been covering in class together. Then there's that video on Bokuila. You don't have to worry about the octopus, but it's just fun to watch if you get bored and want a break. It's a fun video. And then there's the mycorrhizal network chapter with Dr. Simard. Really short video on how trees talk to each other. And then, this TED Talk by Dr. Simard, definitely study this one. Okay. So this How Trees Talk to Each Other, I didn't explicitly have slide sets for that video, but she makes some really good points in there that I want you to think about. And so I will be asking specifically about this video. Not so much about her personal life, that I don't, that's not what I meant. It's the science. So make sure that you are comfortable talking about some of her big ideas there. Oh, and then this plant communication and action. I didn't have a chance to show it today. I was going to show that today, but it's like a 10-minute video basically showing using some of the labeled calcium flow in plants that we saw already, but now they're showing that a plant that's getting chewed on by a caterpillar, and then they video a nearby plant doing that response. And so it's just a way of showing that the VOCs were coming from the plant that was getting chewed on, and you can very visually see how it was ramping up defenses in the non-chewed plant right next to it. So that's a fun video to watch. And then this was the only paper on the sagebrush that I have posted. And so we talked a little bit about that already, but use that as backup if you need to. Questions on exam material? So Monday, we will move forward in the class, topics, but I'll reserve time to go over the exam. I don't have a very solid draft yet. I just gave an exam in my other class that I was fussing around with, so I don't have a lot to tell you yet, other than I'm going to try to make it shorter than the first one. All right. First one had a little bit more, I don't know. There's a lot about what's mycorrhiza and what are endophytes. This one is more talking about the papers that are published and what is the research showing. Okay. All right. So Monday, I'll be able to give you definitely a lot more information on the exam. But certainly email me or set up an appointment. On the weekend, I'll be looking at stuff too. So if you need to reach out on the weekend, I'll be checking email.